And our next speaker is Máté Lengyel, who has a joint affiliation between Central European University in Budapest and the University of Cambridge in the UK. And he's going to be speaking to us about uncertainty in navigation.
Thank you very much. I'd like to thank the organizers for inviting me to this amazing symposium, and it's always lovely to be back at Champalimaud. It's just an amazing place altogether. I'd like to tell you about a project that really grew out of two of my perhaps most important interests over time. A lot of my early career was focused on the hippocampus and on place cells, and in particular phase precession, and then other aspects of hippocampal activities. And then I also became very interested in how the brain might represent and compute with uncertainty. But that work was mostly in the context of perception, where we have amazing paradigms to study this quite directly, both in behavior and at least to some degree in neural recordings, and also to, somewhat later in motor control. But at some point I thought that, why am I having these two lines of research separately, where navigation, which is obviously relevant for the hippocampus, is such an amazingly relevant place to study representations of uncertainty. So that's where this kind of project grew out of, except that on the way, we changed course a little bit, and even though originally I thought that we were going to work on the hippocampus, we ended up in the entorhinal cortex, but who cares about those small differences? As we all know, the hippocampus and the entorhinal cortex, both their discoveries have been awarded Nobel Prizes, and they have been hailed as part of an inner GPS, as being part of an inner GPS in the brain. So, just in case you haven't seen place cell or grid cell recordings, the classical experiments take place in boxes like this. They can be of varying sizes and shapes, and where you record, in this case, for example, from the entorhinal cortex of a rat or a mouse or sometimes other animals. And then what you see if you're recording from just the right place in the entorhinal cortex and you're targeting just the right kind of cells is this amazing pattern of activities, which gives them their names. They're called grid cells because they fire as a function of where the animal is located in space, in this case in two-dimensional space, and their firing fields form a rather regular triangular lattice in space. I'm still, at awe when I look at these kind of results up to this day, because they're just one of the most beautiful examples of kind of something really regular and systematic in neural responses. Nevertheless, there have been some wrinkles on that story, starting from, for example, this paper, but then many other papers have followed, that kind of show that this beautiful triangular or hexagonal, depending on how you look at it, symmetry of grid cells, for which we love them, is actually quite deformed in some cases. For example, in this case, in anisotropic environments. So sometimes they seem to lose this hexagonal or triangular symmetry and do all sorts of funky stuff instead. And so essentially, what this project is about is our attempts to see if there is some method in this madness, whether we can maybe see that these seemingly bug-like features of grid cell responses and corresponding navigational behavior can be actually understood as features rather than bugs. So even though I've introduced my talk with just focusing on grid cell responses, as you will see, a lot of our work is actually targeted at behavioral experiments and not only on experiments in which one can record grid cells. And that's a luxury that this amazing field gives us, because there is so much exciting work going on, and a lot of the times, essentially the same kinds of paradigms are repeated between homing experiments that I'm going to tell you in a little bit, and, for example, rat experiments when you can actually go in and record grid cell activities. So what might be the organizing principle behind these sometimes puzzling behavioral and neural responses? And so to kind of start building a hypothesis, we kind of went back to the basics and thought about what is exactly that a GPS gives you. So you can all, unlike in most talks, I would actually encourage you to all to take your mobile phones out and check that, that this is actually the case, what I'm telling you, is that if you take your favorite navigation app, what do you see here, right? What sort of information does it give you? So obviously, it gives you a map of the environment. That's what it is for, fundamentally. And we know, indeed, that there seems to, that humans and other animals can build rich cognitive maps of their environments. Now, importantly, there is this blue dot there that shows where within that map you are. That's your location signal. And that's obviously very important for efficient navigation. There's also an additional piece of information there, which you might be aware of. Not everybody is aware, so let me tell you that there is that little arrow there, right, that also gives you information about your heading direction. And that, as we just saw in the previous talk, although not only when you're trying to copulate with your partner, but also in other cases, that can actually be a very useful piece of information to know your heading direction. OK, so this is all fine. And indeed, we know an awful lot about the neural correlates of how this blue dot and how this blue arrow is represented in the brain, right? They were worth a Nobel Prize. Actually, the head direction somehow didn't get the Nobel Prize, although some would argue that it really should have. So those are really fundamental and very widely known quantities that are represented in neural activities. But actually, if you look closer, there is one additional piece of information that your GPS gives you that a lot of people actually miss when they look at their phones. But their phones are really using that information not only to display it to you, but also to update their own estimates of your location dynamically as you move. And that is that blue disk around the blue dot. That's your phone's uncertainty about where you are. And indeed, it's not just a gimmick, because under different circumstances, that uncertainty about your location can shrink or grow. So here, for example, I made a sensory ablation experiment with my phone. I switched off its Wi-Fi, and indeed, because it has now one less sensory modality to rely on, its uncertainty has significantly grown. So the basic question for our project was, we know an awful lot about how the blue dot and the blue arrow might be represented in the brain. What can we say about how the blue disk of uncertainty might be represented? How is it reflected in behavior, and how is it reflected in neural responses? And so this is a project, sorry, I'm still learning this pointer. So this is a project that was really very much led by Yul Kang, a very talented postdoc in the group, now has his own group at KAIST, and Daniel Wolpert, a long-term collaborator, who is at Columbia now. OK, so how are we going to formalize mathematically this notion of uncertainty? And I should say, just to manage expectations, that my group is entirely theoretical and computational. We don't do any experiments. We collaborate with people who do experiments, and sometimes we just use the data that they kindly share anyways. So this project is an example for the latter. So how are we going to formalize uncertainty in navigation? So the way we do that is that we are going to build a Bayesian sort of ideal observer model, but rather than ideal observers as they are known in perception, this is going to be an ideal observer model for navigation. So what are the relevant variables for such an ideal observer model? First of all, obviously, the most important quantity is the location of the agent in the environment. Also, it's important to know what the allocentric orientation of the agent is. Now, importantly, these two variables are not observed by the agent directly. There's no magical piece of information given to us. We need to work them out from observations, from sensory information that we actually do have access to. So one form of sensory information is self-motion information. We might have information about our translational movements, and we might have information about our rotational movements. And importantly, as long as we have an internal model or some approximation thereof of how these self-motion signals are actually related to these pose variables, then we might know that depending on how we translate and rotate, that will change our orientation and location over time in successive time steps. We also have other kinds of observations other than just self-motion information. We also, especially if you're a rat, you might have whiskers that you might use to get some information about your environment and whether, for example, you are close to a wall or not. And importantly, and that's going to be critical for this, you have vision, right? You see your environment around you, and that gives you information about location because you know, in principle, as long as you have a halfway decent internal model, how your current orientation and location give rise to these touch or haptic and visual sensor inputs. So this is what we sometimes call the generative model or an internal model, very much along the lines of what Adrian talked about this morning. This is a very simple internal model for navigation. You know that self-motion changes your orientation and location, and that your touch and vision are dependent on your current pose, your current location and orientation. And your job in the world, therefore, is that you need to take a sequence of these sensor observations, and that's what we formalize as an ideal observer model, that you need to take a sequence of these observations and then work your way back to inferring what these latent or hidden variables, orientation and location are. So here's a very simple simulation. We are simulating a rat moving around in a trapezoid arena. These are the visual inputs that we render. And that's going to be critical to the rest of the project. And now we take away the knowledge of where the simulated rat actually is, and we have our ideal observer to actually infer that based on the history of observations about self-motion, touch, and vision. And so at every moment in time, the output of this ideal observer is a posterior distribution. It's actually a posterior distribution jointly over location and orientation, but I'm just showing it over location for simplicity here. That essentially measures the degree of belief that it has that at that moment in time it might be at any particular location. So the red shading here gives you the probability with which this artificial agent believes that they are at any particular location in each of these time steps. Okay, so that's kind of our holy grail. That's what we think any competent navigating agent should be computing over time and update dynamically as it moves around the environment. Indeed, I'm actually in an engineering department. All autonomous navigating agents, be they vacuum cleaners or self-driving cars, use these kind of principles to efficiently navigate through their environments. So this we can compute, and importantly, as I told you, we will, rather than simplifying the visual inputs, kind of abstracting them away to simple scalar measurements, as is sometimes, or in fact very often done in Bayesian ideal observer models, the little piece of innovation along the scaling up theme here is that we actually use an image computable ideal observer model. So we really render the whole image, and the ideal observer really takes into account that whole image. We don't have models anywhere near the sophistication of the kind of models that, for example, Srini is developing based on a complete or an impressively near-complete knowledge of some aspects of the Drosophila visual system. We are just working here still with a relatively simplified kind of cyclopean vision model, but already this gives us interesting insights that would have been impossible to get with just standard abstract Bayesian ideal observer models, as it turns out. All right. So I told you that the key object that this model computes in every time step is the ideal observer's posterior. And I'm sorry again, I'll have to go back in a rather tedious way. I'm trying to make this quick, because there is one thing that I would like to draw your attention to, and that's going to be critical, which is that this posterior distribution roughly follows the true location of the animal, as it should, if it's a reasonable ideal observer model, if it's really ideal. But importantly, as you can see, the overall uncertainty it expresses by the spread of this posterior distribution changes dynamically from moment to moment. And moreover, it's not only changes in overall magnitude, it can also sometimes become quite anisotropic. Sometimes it becomes elongated in one direction and much narrower in some other directions. So it really is very dynamically modulating its spatial uncertainty as it goes along. All right, so how are we going to extract actual experimentally testable predictions from this abstract object of a posterior distribution that is computed by some ideal observer. So first of all, we are going to make contact with human homing experiments and human homing behavior in those experiments. So those experiments essentially have two phases. In one phase, the familiarization or the learning phase, humans find an object somewhere in the environment, and that we are going to model with our ideal observer. It's going to follow the same sort of trajectory in our simulation, and at every moment in time, it computes a posterior distribution about where it is. When it finds the object, it will, that's the posterior of interest, and it will store in memory, as the location of the object, it will store in memory the mean of that posterior distribution that it computed at the time when it found the object. Now, during the test phase, the object is removed, and the human needs to find its way back to where it thinks the object should be. That's the basic task in the experiment. And we are going to model this in a way that we also restart our agent from somewhere else in the environment. We might add some noise just for good measure to the representation in the memory representation of the previously memorized object location. And then we are going to simulate an agent that, again, at every moment in time, it computes its posterior distribution over where it might be in the environment. And then, so here it is. For example, at this point, it computes a posterior that it's here, and it will follow a trajectory that is going to try to minimize the expected square error between its current belief about where it is and its belief about where the object should be. And so it will stop when this expected or mean squared error is minimized. Why is that important from our perspective? Because when you minimize mean squared error, you don't just want to match the mean of the posterior to the memorized location, because the mean squared error, as we all know, can be decomposed into a bias term, which indeed is just the distance between the mean posterior and the remembered location. But there is also a variance term, which is exactly your uncertainty about where you are. So that already suggests us that these dynamically modulated uncertainties should therefore influence homing locations, and we should be able to see their signatures in behavior if all is well. Okay, so that's going to be how we make predictions about behavior in these homing experiments. Next up is the way we are going to turn our ideal observer into predictions about neural responses of grid cells. So again, here is a simulation of a simulated rat. We are computing ideal observer posteriors for this rat based on kind of simulated sensory inputs that it receives as it goes along this trajectory. So these are, again, the ideal observer posteriors, and as you can see, again, they are roughly tracking the true position of the animal, but again, there is some interesting modulations of uncertainty, both in overall magnitude and in overall shape. Now, we are going to consider a population of grid cells at different scales and different phases, as usual. We are going to start from the assumption that they have roughly hexagonal symmetry. So we are not going to explain why they have hexagonal symmetry, unlike a lot of work that is focused on exactly on that aspect. We are starting from hexagonal symmetry, and we will be interested in how that hexagonal symmetry might be deformed and why. So if we start with a population of grid cells with entirely regular grid fields, then we can ask what sort of representation does such a population give us about the location of the animal? And so that we can compare it to ideal observer posteriors, what we are going to do is what actually people in the field do a lot of the time when they analyze their own recordings. They construct what they call a Bayesian decoder of location based on neural responses. So they ask, if I don't actually know the location of the animal, I'm just trying to figure the location of this animal out based on the neural responses of a population of grid cells, say, what would be my best estimate? And that best estimate is going to be probabilistic, because I can't really be sure where I am based on these neural responses, and I can plot that again at every moment in time. And so if we do that, and use such a kind of a Bayesian probabilistic decoder, using a population of grid cells with regular grid fields, then we get posteriors that roughly, again, track the true location of the animal, and therefore roughly track the ideal observer posterior, which kind of quantifies the exact amount of uncertainty that an ideal observer should have at that point. But what you see is that these neuroposteriors are not really modulating the uncertainty, right? So at every moment in time, except for trivial boundary effect, these neuroposteriors that regularly tuned grid cells can represent do not show the interesting and important modulations of the ideal observer's posterior. So what we do next is that we start deforming these grids. We warp the grids jointly. And we optimize the warping of these grids so that now, if we use a probabilistic decoder on these warped grid cell responses, then the ideal observer posterior is well matched by what we call these neural posteriors. The match is still not perfect, as you can see, but it's much, much better, and we can quantify it. It's much better than with regular tuning curves. So importantly, we compute that optimal warping once for the whole population, and that single warping should result in neural responses that match the ideal observer's posterior from time step to time step, right? So that's a very kind of highly constrained optimization problem. And again, we are indeed not doing a perfect job, but we can do a reasonably good job at that. OK, so that's the basic idea. So essentially, our predictions about grid cells is that their grid fields are going to be distorted exactly in the way that would allow them to represent exactly the right amount and form, or if you like, shape of uncertainty at every moment in time. OK, so unlike efficient coding approaches, the objective here is not to represent as little uncertainty as possible, because that would be fooling yourself, because you cannot have absolute certainty about your location during navigation to begin with. That's exactly what our ideal observer model quantifies. Instead, the objective is that your grid cells should respond in a way that represents just the right amount of uncertainty at every time point. All right, so that's our prediction for neural responses, the optimized tuning curves. OK, so this is a complicated model, and I'm going to give you kind of a whirlwind tour of all the kind of phenomena that it reproduces, with essentially 2 free parameters. But in order to actually get some deeper analytical insight, we can develop at least some simple back-of-the-envelope calculations for what might be the principles behind the operation of this rather complex image computable model. And so what we came up with is that we can understand an awful lot from the operation of this model by just considering this very simple setting of an animal being in a totally kind of almost being in a vacuum with just a simple single point-like landmark. And the question is, how much information does this agent have about its location, for example, along this coordinate, the d coordinate, the distance coordinate, when its true distance is d and the eccentricity of the landmark is the distance along the line of sight is d, and the eccentricity is epsilon. And so you can do a simple back-of-the-envelope calculation and show that this information scales inversely with the distance, and in fact with the 4th power of the distance, and with the square of eccentricity. Okay? So that's interesting, because that already tells us that we will see interesting interactions as the animal moves around, because now the information that it's getting from vision is going to be dynamically modulated based on its distances from different landmarks in the environment. We are not simulating discrete landmarks. We are really simulating the whole bounding box of the animal. But this is a good approximation, it turns out, to that. And just to show you that, indeed, if we simulate the full model, this, with just a single landmark at least, this, analytical formula describes the actual visual information that we can extract from the model pretty well. All right. So keep that result in mind, because that's going to be, underlying a lot of the complexities that we'll see later. So first of all, we can simulate what happens in a simple square box, and you can see that the visual information that is driving a lot of the final spatial uncertainty is more or less homogeneously distributed with some inhomogeneities and obvious symmetry, along the x and the y-axis. Whereas if you are in a trapezoid, there is much more anisotropy going on, and there is also much bigger differences about x and y. Importantly, if you kind of add up these, information around X and Y in just the right way, then you can find that overall there is much more information in the square than in the trapezoid. The total information in the square is really quite homogeneous, whereas the total information about location is much more heterogeneous in the trapezoid. So now we can go to these homing location homing experiments, and see what happens in those homing experiments when we measure people's spatial memory. And indeed, we find that the memory score is much higher in square than in trapezoid environments. Well, we don't find it. These people find that. It's much higher. Their memory seems to be much better in the broad side of the trapezoid than in the narrow side of the trapezoid. And indeed, the model reproduces that essentially based on these principles, and we can really understand these spatial information maps to a very good approximation based on the simple formula that I just showed you on the previous slide. Okay, I don't really have time to go into it, but it turns out that the same model also explains some slightly more surprising aspects of these experiments. And in particular, here, people were asked to estimate distances between different locations. And while it makes an awful lot of sense that when the memory score is better, their estimation errors or biases are smaller in some cases, like here, in absolute magnitude. There are also some puzzling effects here, whereas the memory score is better in the broad side of the trapezoid than in the narrow side, and yet the overall magnitude of estimation errors is bigger in the broad side than in the narrow side. And it turns out that with a little bit of more thinking, we can understand why that might be the case, and indeed the full model reproduces that behavior. So these simulations are always from the full model, and then we can do some back-of-the-envelope calculation to understand why this is happening in the model. All right, in the interest of time, I'm just going to show you the corresponding results. So this is what I told you, that one of the beauties of working in this field is that there are a lot of parallel experiments that use almost the same sort of manipulations for human behavior as for rat navigation. So this is the famous experiment of Julia Krupic in John O'Keefe's lab, and they showed that the gridness of grid fields in the trapezoid environment is seriously diminished, and in particular in the narrow side more so than in the broad side, and likewise the diameter of these grid fields also grows in the trapezoid, and especially in the narrow side more so than in the broad side. And again, the model reproduces that, at least qualitatively, quite well. There is a host of other phenomena that the model reproduces that I don't really have time to tell you about in detail. Look at the preprint if you're interested, or talk to me after the talk. There's one more phenomenon that I wanted to very briefly mention, and that's tethering, if I ever get there with this clicker. Okay, so as you can see, there's a whole host of experiments where similar manipulations have been done with humans and rats, and we reproduce, at least qualitatively, all those results with a single model. I wanted to tell you about this sort of experiments when subjects are in a box. They need to, again, think of a homing experiment in this case. The subject is driven to a particular location that they need to memorize, and then unbeknownst to them, the environment, and importantly, in that environment, they can make inferences based on where they are, based on essentially two kinds of cues along this axis, cues that are tethered to the starting wall and cues that are tethered to the opposite wall. And in the original environment, of course, they will agree on where the object should be. But now, and so the combined estimate is going to be there. But now, if you, unbeknownst to the subjects, you extend the environment, maybe not so dramatically as this makes you believe, but this is just an exaggeration, then these two kinds of cues are not going to agree anymore. And so now, this is where it really becomes important that we understand how visual information, but also self-motion information, scales with distance from these different walls, because if we actually think about it, we can already understand why, depending on the original target location, one or the other kind of cue is going to dominate, and therefore the combined estimate that combines all these cues together is going to be shifted towards the starting wall or towards the opposite wall, and therefore why overall we will see a stretching of responses, but also we can can understand why overall the shifting towards the starting wall is bound to be stronger simply because there is more and better cues that tether responses to that starting wall. And that, again, is provided by kind of a mathematical analysis of the model. Okay, so that suggests that overall there is going to be a net shift towards the back wall, which has been called tethering, this phenomenon that has been observed both in behavior and in neural responses. And indeed, therefore, the full model reproduces both tethering in human responses and in the grid-cell responses of rats in similar experiments. Okay, and so with that, I'd like to summarize. I told you how uncertainty is absolutely inherent in navigation. I told you how, by constructing an image computable Bayesian ideal observer for navigation, we can actually quantify that uncertainty, how that ideal observer gives us behavioral as well as neural predictions, and how that explains memory and grid field deformations, as well as tethering, and a host of other phenomena that I didn't have time to go into. And with that, I'd like to thank my two amazing collaborators on this project, Yul and Daniel, and generous funding by the Wellcome Trust. Thank you very much.